Hi guys, Professor Ruggieri here. I'm about to start the setup for the water activity osmotic pressure lab, and I just wanted to give you guys a walkthrough of what I'm actually doing rather than making it sort of idealized. So I currently have both the S. epidermidis and E. coli cultures that I rehydrated, rather, on Friday. So I'm just gonna put this down for 1 second. Let's see. I want to tighten the lid on this guy right here, and I want to agitate it a little just to make sure that it is in fact cloudy, right? And you can see that it is. You can't see directly through it. It's cloudy. A lot of the stuff had sedimented to the bottom. Same thing with the E. coli over here. And agitate it a little, and you can clearly see that it's cloudy, right? So there's confirmed growth in both of these. And if you want a comparison, just look at an uninoculated nutrient broth or tryptic broth tube, and it'll be clearer. You also want to get out this package right here. Okay, so I'm going to open this up, and I do apologize. I'm filming this all by myself today, so I'm going to pause this, then merge it with the next video. Basically, if you look, these are all vials of broth with different saline, different salt concentrations, therefore different osmotic pressures. So there's 4 sets, okay? And if I go over, just to save paper, I'm doing this on my tablet here. Oops. Let's see. We have the protocol pulled up, right? So we're gonna be doing a tube of 0% sodium chloride, which is just broth, okay, regular broth, for E. coli and for Staph epi. Then we're going to be doing a tube of 5% sodium chloride, 10%, and 15%. That means that if this is the 0 set, this is the 5, this is the 10, and this is the 15, the first thing I need to do is take these out of the package, they're already at room temperature, and write on the label, okay, either S. epi or E. coli, and then my initials and the date. So when I come back in the next video, I'll have that all set up. Okay, everybody, I just want to walk you through my little setup here before I move on to the last stage, which is going to be inoculation of all these broth tubes. The first thing I want to point out is just the relative clearness, right, of the uninoculated. In this case, it's the salt broths compared to the inoculated either S. epi or E. coli broths. You can clearly see the difference between them. Okay, the other thing I did was I slightly loosened the caps on the Staph epi and the E. coli tubes to make them easier to work with. And then each of these sets of sodium chloride broths, these lids were on insanely tight because they were packaged in the box, right? They weren't supposed to spill. So I loosened those. What you'll also notice is I did put my initials and the date on one of these because they're a set. So you don't really need to put your initials and the date on every single one. But I lined it up, so here's my broth culture of Staph epidermidis, and then I have all my S. epi tubes in order of increasing salinity, right? So this is the 0%, this is the 5%, this is the 10%, and this is the 15. And I did the same thing for the E. coli in the back, okay? Organization is key, and you have to label everything before you get started. If you don't label, you're going to finish doing this and not have any idea what you inoculated with. Now comes the easiest part, right? We're just going to take a loop full of each of these and put it into each of these tubes, and we're going to wait 48 hours. So I'll show you how I'm going to do that. Okay, guys, so I'm going to attempt to light my flame here. Don't mind my dirty t-shirt, I was painting, okay? If this goes out, no big deal. Just make sure, the most important thing here, after you've labeled and everything, is that you have your disposal container handy so that you're not just left in your kitchen with bacteria-ridden loops, okay? Now, if you're careful, you can actually use the same loop. Well, actually, I take that back. That's not a good idea. It's not good practice because each of these broths has a different salinity, so you don't want to contaminate them. So we're going to use a different broth into each of the different tubes, okay? So here I have my sterile loop. I'm going to actually tighten the lid on the S. epi tube a little bit and just agitate it to mix it up. Then I'm going to open it. I'm going to flame it here. Okay. I'm going to dip in, get a nice loop of bacteria, flame it again, put the lid on it, put it back. Okay, then I'm gonna go into my 0% sodium chloride broth, flame the loop, okay? Put that down inside, oops. Drop this in here, and I'm about to drop the lid, so I apologize. Flame it if the flame was still going, and put the lid on, maintaining a little bit of shakeability there, okay? You can actually make sure all these lids are a little bit loose to make it easier. So I'm gonna do the same thing 3 more times, right? I'm gonna take my tube of Staph Epi, run it through the flame, dip the loop in there, flame the lid, cap it, put it back, go into the 5% sodium chloride, put the cap back on, and you're supposed to be flaming the sodium chloride broth, but honestly, given how weak the flame is, if you just go in and out quickly, you should be okay. So 2 more, right? I'm going into the 10% sodium chloride. I get a big dollop, a big loop full of my Staph Epi, flame it, put it back. Take the 10%, if you want to flame it, you can. If not, be super quick, open the lid, put the loop full of bacteria in there, close the lid, dispose of your tool. Now I have my last Staph Epi tube, which is the 15% sodium chloride. Flame the lid, flame the mouth rather, get a loop full of bacteria, flame it again, put the lid back on, take my 15% and go right in with the loop. Okay, cap it and put the loop back in. And I'm gonna do the same exact thing for my E. coli, right? And after I've done it for E. coli, remember, don't get rid of these broths. It's the same broths that you're gonna need, this broth culture, you're gonna need it for the pH lab and for the selective and differential. So if you had everything, if you had Serratia marcescens, Micrococcus luteus, E. coli, and Staphylococcus epidermidis, then you